Thanks so much for this opportunity to present some of our recent work and generally the state of the field, understanding immune responses to cancer immunotherapy from outside of the tumor and how those really impact therapeutic efficacy. My name is Matt Spitzer. I'm an associate professor at UCSF and the Gladstone Institute's an investigator of the Parker Institute and the Chan Zuckerberg Biohub. A few financial disclosures. I'm a shareholder and co-founder of Takeo Bio, which is a company that provides immune monitoring services. And I've also received research support from Genentech, who's the manufacturer of atezolizumab, which will be discussed in the presentation today. So in general, one of the questions that really fascinates us is thinking about how the immune system makes decisions. And this can happen at multiple levels of regulation. At the organismal level, the immune system is broadly responsible for regulating physiological homeostasis and the response to any breaches in homeostasis. At the tissue level, immune cells constitutively infiltrate every tissue across the body, and these cells distributed across large anatomic distances need to coordinate their responses when an immune response is mounted in order to form a coherent immune response. And at the cellular level, there are many different types of cells, each of which have different roles and responsibilities, which again constantly communicate in order to drive cellular decision making. There's no master regulator cell in the immune system that provides instructions to everyone else. This really happens collectively through continuous communication amongst immune cells. So the question I'll focus on today is, where do T cells get activated after cancer immunotherapy? We've come to appreciate that there are T cells that infiltrate into tumor microenvironments. If there are many T cells, we call these hot tumors. If there are few T cells, we call these cold tumors. But stereotypically, or often what we think about during immune responses is that T cell responses are initiated in secondary lymphoid organs, including in lymph nodes, which receive fluid through the lymphatic system and through which cells migrate from tissues in order to survey whether there are any foreign molecules present in those tissues by presenting antigens to T cells and B cells that reside within these lymph nodes, as well as the spleen. And when an immune response is initiated, T cells will get activated in the lymph nodes, proliferate, and eventually egress into the circulation, through which they'll arrive back at the tissue in which there's damage or infection or cancer, extravasate through the vasculature, enter into the tissue, and drive effector function within the tissue. And so really the question that we've been interested in understanding is, during effective cancer immunotherapy, are we just reactivating T cells within the tumor microenvironment, or are we driving these broader systemic responses in which T cells from outside of the tumor get activated and are participating in the response itself? Our interest in this area stemmed from a study that we did a number of years ago at this point, where we investigated the mechanistic basis for effective immunotherapy. We started in a mouse model of breast cancer, the polyomma T model that spontaneously develops breast cancer, and we left mice either untreated or treated them with an effective immunotherapy consisting of tumor-binding antibodies and dendritic cell adjuvants. And at two different time points over the course of the immune response, we collected all of the different tissues that we thought might be involved in this response and analyzed them at the single cell level to understand immune cell composition as well as behavior and functionality using mass cytometry. So just a brief overview of mass cytometry. This is a technology that allows us to measure many proteins at a time in single cells. We start with a single cell suspension, we can exclude dead cells from our analysis using a viability stain, and we can multiplex samples together by attaching barcodes to each individual cell that allow us to delineate which sample a cell came from. So we can pool many samples together, stain them with antibodies conjugated to heavy metal isotopes. So each antibody is tagged with a different metal isotope that has a unique molecular weight. And then we can analyze those cells one at a time. The nebulizer will put these into little droplets. They get carried into an inductively coupled argon plasma that will ionize the cell, break the covalent bonds in the cell to produce an ion cloud. We filter the ion cloud using a quadrupole to get rid of all the low molecular weight carbon, oxygen, nitrogen that comprises the majority of the cell. and enrich this heavy metal ion cloud. And then we can quantify the metal ions on each individual cell using a time-of-flight mass spectrometer. And at the end, we result in a data matrix where each row is an individual cell that was measured, and each column is the expression value for a given protein in that individual cell. And we can go about many different ways of analyzing the data using conventional approaches as well as high-dimensional approaches. And so what we observed in this initial data set, asking what happens during effective immunotherapy, is that there was broad immune cell proliferation taking place in the draining lymph node, but not in the tumor microenvironment at the time that the tumors were actively shrinking and being rejected by the immune system. So what's visualized here are dimensionality reduction plots, a method called scaffold maps, where each node in this graph represents a cluster of immune cells that has similar protein expression profiles. And each cluster is colored based on whether it changes in its rate of proliferation by measuring the proliferation marker Ki67. And so on the left side, we see in the tumor microenvironment that there's no significant change in immune cell proliferation rate with effective immunotherapy or ineffective immunotherapy. Whereas on the right side, we see a dramatic increase in proliferation across many immune cell clusters in the lymph node at this same time point. So that really led us to ask the question, are these responses from outside of the tumor important for driving anti-tumor immunity and efficacy? And the way that we went about testing this, or one way that we did, was to block the egress of immune cells from the secondary lymphoid organs, such as the lymph nodes. So we asked if we prevent cells from leaving the lymph nodes at the time that immunotherapy is given, will it still be effective? And we did this using a molecule called FTY720 that blocks the sphingosine-1 phosphate receptor on immune cells, preventing them from leaving the lymph nodes. And what we found is if we blocked egress, we lost the efficacy of this immunotherapy in our model of breast cancer. We then asked, is this more broadly generalizable to other types of immunotherapies? And so we turned to a mouse model that is responsive to checkpoint inhibitors, called MC38. This responds pretty well to anti-PD-1 immunotherapy. And we saw the same result. If we gave FTY720 together with anti-PD-1, we saw that the tumors continued to grow rather than being controlled by the immune system, suggesting that immune cells from outside of the tumor were important contributors to the success of the immunotherapy. And similar results were published right around the same time from a couple other groups as well. Now, I'd like to take a brief pause and review some work that was happening around the same time to understand the differentiation state of T cells in the context of cancer. So when naive T cells are activated, say in the context of an acute infection, they'll typically differentiate into effector cells that can kill infected target cells really well as long-lived memory cells, which provide long-term protection from recurrence of whatever that antigen was. However, in the context of cancer as well as chronic infections, the antigen doesn't get cleared, and so T cells are subjected to continual stimulation through the T cell receptor. And this leads T cells to differentiate in a slightly different way into a program that's been termed T cell exhaustion. So these T cells ultimately downregulate their effector function in order to prevent immunopathology. And as a result, they make less cytokines, they're less proliferative, and they're not as good at killing target cells. Now, it's been discovered that within this exhaustion differentiation trajectory, there are early exhausted cells, which are oftentimes called progenitor or precursor exhausted cells. These cells express a transcription factor called TCF1 that's associated with proliferative capacity and self-renewal capacity. And when these early exhausted or progenitor exhausted cells get activated, they differentiate ultimately into terminal exhausted cells that have downregulated their effector function as well as their proliferative capacity. And it's been shown in a variety of systems by several groups that these early exhausted cells are really responsive to immunotherapy, whereas the terminally exhausted cells have undergone stable epigenetic modifications that prevent them from being reactivated easily, and these are generally not responsive to immunotherapy in the same way. Work from other groups, including Howard Chang's group at Stanford, identified that in human patients treated with checkpoint inhibitors, new T cell clones infiltrate the tumor after treatment, and these new T cell clones tend to be in this exhausted state that's associated with tumor protection. So this was really elegant work done through single-cell RNA sequencing together with paired T-cell receptor sequencing in order to look at the T cells and the T-cell receptor sequences on those cells present prior to treatment and after treatment in patients with basal cell carcinoma. And the authors found that many new clones are activated, identifiable in peripheral blood, and infiltrate the tumor newly after treatment rather than the clones that were pre-existing within the tumor becoming reactivated. So this was very consistent with our data from our mouse models. In addition, several other groups around the same time identified that in the peripheral blood, there's an expansion of new T-cell clones that belong within an effector-like T-cell state. In this particular study, the authors defined these cells as being CD45-RO positive and negative for CCR7 and CD27. And they found that expansion of these effector-like cells in peripheral blood took place to a larger extent in patients with disease control, as opposed to those who had progressive disease after immunotherapy treatment. And they showed that expansion of this particular subset in peripheral blood was also strongly associated with overall survival in this cohort of patients, suggesting that these responses detectable in the periphery are really important and strongly associated with clinical benefit from immune checkpoint inhibitors. Moreover, work done around the same time in mouse models really started to delineate the types of CD8 T cells that get activated and respond to checkpoint inhibitors. So elegant work done by Dammeier et al. Found that T cells within tumor-draining lymph nodes get activated after immune checkpoint inhibitor treatment, particularly by blocking the interaction between dendritic cells that express PD-L1 in the lymph node and T cells that express PD-1 in the lymph node. These cells then clonally expand and ultimately egress from the lymph node into the blood, infiltrate into the tumor, and carry out their effector functions. And very nice work from Ira Mellman's group at Genentech came to a similar conclusion as well, reinforcing the notion that these immune responses from outside of the tumor are critical mediators of immunotherapy success. And lastly, I'd like to highlight some work from the Joshi and Jack's labs that identified a reservoir of these stem-like or progenitor-like CD8 exhausted T cells that reside in the lymph nodes. So using some elegant mouse genetics systems here, the Joshi lab was able to find that there's a population of PD-1, TCF1 positive antigen-specific CD8 T cells that reside in the lymph nodes, and that, again, by blocking the trafficking of immune cells out of these lymph nodes, you eventually saw a depletion of antigen-specific cells in the tumor microenvironment while these cells were retained in the lymph node, suggesting that the cells from the lymph node are important for replenishing the pool of CD8 T cells within the tumor microenvironment. So just to summarize some of these observations, immune responses outside of the tumor contribute to the efficacy of immunotherapy. Studies have shown that T cell clones expand in the periphery after treatment and infiltrate into the tumor, that CD8 T cells in the draining lymph node respond well to checkpoint inhibitor immunotherapy, and that there's a specific population of these progenitor exhausted CD8 T cells that reside in the draining lymph node, particularly in mouse models. And so from there, I'd like to start talking about work that we've done more recently to evaluate to what extent this is true in patients and whether the same types of CD8 T cells are responding. So this was just recently published in Cell. And what we did was to take patients with head and neck squamous cell carcinomas and to analyze paired tumors and lymph nodes from these individuals. And we did this using mass cytometry, using an antibody panel oriented toward T cell differentiation states. We performed dimensionality reduction on all of the CD8 T cells and identified clusters of CD8 T cells that express shared protein expression profiles, and then annotated these based on their differentiation state as indicated here in the heat map. And we found populations of progenitor-exhausted, terminally exhausted cells, as well as the more conventional differentiation states, naive central memory, effector memory, and tissue-resident memory cells, and asked which of these is more or less abundant in the lymph nodes compared to the tumors from these paired samples from the same individual. So we performed a differential abundance analysis on these clusters and identified that many of the terminally exhausted clusters were enriched in the tumor. And in contrast, the progenitor exhausted clusters, 14 and 8, were enriched in the uninvolved lymph node. So these formed a larger percent of the antigen-experienced T cell pool within the uninvolved lymph node than they did in the tumor, as quantified on the right. These were highly statistically significant. We then wanted to know, are these progenitor exhausted cells in the lymph node clonally related to cells that we see in the tumor microenvironment? And so we analyzed CD8 T cells from 5 additional patients by single-cell RNA sequencing paired with T cell receptor sequencing. We identified the CD8 T cells, clustered them, and colored them in this UMAP dimensionality reduction plot based on their tissue of origin, identifying regions that were enriched in lymph node or tumor respectively. And we overlaid onto this gene signatures from a recent meta-analysis of CD8 T cells across many different human cancers published by Zheng et al. And we're able to see regions that were enriched for the Tpex or TCF7 positive populations compared to the terminally exhausted populations, which were enriched over on the right, where cells from the tumor microenvironment were also preferentially enriched. We then identified clones based on their shared T cell receptor sequences, and were able to identify across each patient clones that were shared between the lymph node and the tumor. And we asked what were the differentiation states of these CD8 T cells that belonged to these shared clones across each tissue. We found that the cells in the lymph node were more likely to express the TCF7 Tpex-like exhaustion score, so progenitor exhausted cells, whereas their counterparts with the same clones, same T cell receptor sequences belonging to the same clones, were enriched in the terminally exhausted cells in the tumor microenvironment. And plotting this on a clone-by-clone basis, we also saw that the cells in the lymph node had overweighted Tpex gene expression signatures relative to the terminally exhausted signatures compared to the counterparts that belonged to the same clone but were in the tumor microenvironment instead. Now, of course, we wanted to know, do these cells respond to checkpoint inhibitor treatment? And so here we, together with our clinical collaborators at UCSF, performed a small neoadjuvant clinical trial where patients with head and neck cancer were treated with a single cycle of atezolizumab anti-PD-L1 shortly before surgery. And in the context of this study, we were able to look at what happens in the lymph nodes after patients are treated with this checkpoint inhibitor, and we compared it to lymph nodes collected from 22 patients receiving standard of care surgery without immunotherapy. We identified regions in these lymph nodes and put them into a tissue microarray that were analyzed by multiplexed ion beam imaging. And multiplexed ion beam imaging is a technology related to mass cytometry using metal conjugated antibodies to quantify proteins. But instead of doing this on a single cell basis on cells in liquid suspension, it instead uses a primary ion source to liberate these ions from antibodies that have been used to stain intact tissues, intact tissue section. And so this allows us to see where physically within the tissue each cell is localized. And it's compatible with formalin-fixed paraffin-embedded tissues that are oftentimes the way that tissues are processed for clinical pathology applications. And so we did the same thing from our MIBI data. We identified all of the CD8 T cells, and we annotated them based on their differentiation states, based on the proteins they express, and found that a population of Tpex was present in the lymph nodes, as well as some more intermediate exhausted cells that had downregulated expression of the transcription factor TCF1, but had not yet upregulated the expression of inhibitory receptors like TIM3 or CD39 associated with a terminally exhausted state. And we asked across all of these CD8 T cell clusters, how do they change after treatment with immunotherapy? And we found that the progenitor exhausted population, cluster 18, was the most significantly reduced in patients that had recently been treated with anti-PD-L1, whereas cluster 16, this intermediate exhausted population, was amongst those that had the largest increases in their abundance within these lymph nodes. So we quantified this more directly, and we found a strong trend toward a decrease in Tpex in immunotherapy-treated lymph nodes from patients. as well as a trend toward an increase in the intermediate exhausted cells. And if we express this as a ratio of the PD-1 positive CD8 T cell population, we could see a significant skewing of this population toward the more differentiated intermediate exhausted state as opposed to the progenitor exhausted state. And because this was spatial data, we were also able to look at which cells are physically neighboring Tpex in the tissue, and found that Tpex after treatment were surrounded by larger numbers of these intermediate exhausted CD8 T cells. We then asked, do Tpex co-localize with any antigen-presenting cell populations that may be driving their activation after treatment? And indeed, we found that after treatment, Tpex were more likely to be surrounded by larger numbers of dendritic cells compared to patients that did not receive immunotherapy. And importantly, the total number of dendritic cells, or the percent of dendritic cells in these tissues, was not changed by treatment. So this really reflects a change in the localization of these cells. And moreover, we saw that the Tpex, as well as the intermediate exhausted cells that were neighboring dendritic cells, upregulated the expression of PD-1 in the lymph nodes from patients treated with checkpoint inhibitors, consistent with the activation of these cells. Now, as I mentioned, several studies had found that expansion of particular effector CD8 T cell populations in peripheral blood was associated with clinical benefit from immune checkpoint inhibitors. And so we were interested in understanding how these responses that we observed in the lymph node related to responses we might see in peripheral blood. So from the same patients, we collected pre- and post-treatment blood samples, which were analyzed together with some resected tumor tissues by mass cytometry. We identified the CD8 T cell clusters, subjected them to clustering, and annotated the identity of each cluster based on the expression profiles of the cells within that cluster. And what we found was that comparing the pre-treatment time point, baseline, to the post-treatment time point at the time of surgery after a cycle of checkpoint inhibitor immunotherapy, that there were two clusters of intermediate exhausted cells with similar phenotypes to those we saw expanding in the lymph node that also expanded in peripheral blood. We also saw that one of these clusters, cluster 5, significantly increased its proliferation in peripheral blood as measured by a Ki67 expression. So these cells had recently or were in the process of dividing. And we also saw this to be true of a TEMRA population in the peripheral blood. Now, it was possible that these cells in the blood originated from the lymph node CD8 T cells that we saw responding, or could have potentially egressed from the tumor. And so we asked, do we see evidence of similar populations proliferating in the tumor? And we did not. So these cells were Ki67 positive in the blood, but not in the tumor tissue, which would be consistent with their etiology as having come from the lymph node. And so this was really interesting to us because it really drove home this point that the responses we see in the lymph node are indeed associated with these detectable responses in peripheral blood, which is a great opportunity for immune monitoring. Now, the last question we wanted to address in this study is what happens in metastatic lymph nodes? Cancer cells can metastasize to regional lymph nodes, and we wanted to know if that impacts the responses of CD8 T cells within the lymph nodes when immunotherapies are used to treat patients. And from a number of patients, we were able to collect paired, uninvolved, and metastatic lymph nodes from the same individuals at the same time, draining the same primary tumor. And we asked what CD8 T cell clusters were differentially abundant between them. Now, these metastatic lymph nodes really looked a lot like the primary tumors. So they had a significant reduction in the abundance of these Tpex clusters as compared to the uninvolved lymph nodes from the same individuals. And we then asked whether these changes in the lymph node would extend to the response to immunotherapy. So we investigated metastatic lymph nodes from patients treated as part of the clinical trial, and we found that indeed, in metastatic lymph nodes, progenitor exhausted cells failed to be surrounded by larger numbers of intermediate exhausted cells, consistent with their lack of differentiation. And the Tpex and intermediate exhausted cells also failed to upregulate PD-1, consistent with the lack of activation of these cells in the metastatic lymph nodes compared to the uninvolved lymph nodes. Then we were curious what cell types these cells may be interacting with within the metastatic lymph nodes, and if they had a different phenotype. And indeed, we found that the dendritic cells surrounding the Tpex in these metastatic lymph nodes had upregulated many immunosuppressive molecules, including CD39, IDO, PD-L1, and TIM3, as compared to the dendritic cells from the uninvolved lymph nodes of recently treated patients. And moreover, we found that the Tpex were also surrounded by Tregs that had much higher FOXP3 expression and higher expression of TIM3, which are both consistent with the suppressive capacity of Tregs within these metastatic lymph nodes. And this is visualized in the representative image here at the right. So having seen that the response to checkpoint inhibitors is impaired in metastatic lymph nodes, we then wanted to know, is this then associated with a change in the detectable CD8 T cell response in peripheral blood? And indeed, that was what we saw. So patients with metastatic lymph nodes tended to have smaller increases in these intermediate exhausted cells within the lymph node and less proliferation of these cells after treatment. And so just to summarize the findings of our new study, what we've found here is that Tpex are indeed present in regional lymph nodes in human cancer patients, and these Tpex are clonally related to terminally exhausted cells that are present within the tumor microenvironment. We found that immune checkpoint blockade reduces Tpex abundance in these uninvolved lymph nodes, but increases the abundance of intermediate exhausted cells. We found that checkpoint blockade also results in Tpex localizing near DCs where they become activated. And we found that in metastatic lymph nodes, Tpex are both reduced in frequency and localized in immunosuppressive niches that prevent them from getting activated well when patients are treated with immune checkpoint inhibitors. And so with that, I'd really like to acknowledge everybody in the group that contributed, especially the first authors of the study, Trainalina Okholm, a fantastic computational biologist in the lab, Kyle Jones, a pathologist, and Maha Rahim, an exceptional experimental biologist. This was really a team effort that involved many people throughout the lab. Our clinical collaborators, Alan Al-Ghazi, who's the medical oncologist who treated these patients, and the PI on the clinical trial, Patrick Ha, the surgical oncologist who operated on all of these patients, Larry Fong, the director of our cancer immunotherapy program at UCSF, and the driver of many of these immunotherapy studies, Mika Van Zant, our fantastic collaborator in pathology, who really helped us access these lymph node tissues in a way that was consistent with and commensurate with the clinical pathology that needed to happen, Max Krummel and Alexi Combs, who were instrumental in the single-cell RNA sequencing studies, Stanley Tamaki, instrumental in the MIBI studies, Alex Chung, instrumental in study logistics, sample processing, collection, and Bushra Samad and Vrinda Johri, who helped with the single-cell RNA sequencing studies as well. Our fantastic collaborators at Stanford, Mike Angelo and Candice Liu, who helped with the MIBI analysis, Bill O'Gorman at Genentech, who was instrumental in setting up this program, and of course, most importantly, all of the study participants and their families, without whom and without whose participation and support and partnership, we would never be able to do these types of translational research studies, as well as all the funding sources that make these types of studies possible. Thank you so much. Particularly to Genentech and the Cancer Research Institute for their partnerships in this study, as well as the Parker Institute for Cancer Immunotherapy. And thank you so much for your attention. Take care, everybody.